Here's Bill Henderson.
The flash reader and our new food safety testing system can be seen as a disruptive technology to the food industry and food safety testing. Our synthetic DNA assay system can provide an opportunity for a great return on investment to investors. I can tell you that from my years of experience in cost and financial models and operations management, and as an investor. Before we developed our first prototype, we made presentations to testing lab managers, food processing companies, and potential investors. The comments were, if you can develop such a system, we want it. And investors, including some VC investors, said, come back to see us when you have it. We're interested. It has not always been easy, as many of you will attest to, when you are developing an innovative technology and wanting to start up a new company. We were not successful in our first attempt to get funding from the Texas Emerging Technology Fund, and one comment stuck with our team. Can you do a demo to show how it works and that it does work as you say it does? We then knew what we had to do. We developed our first prototype assay for Campylobacter and did numerous lab tests to optimize the assay to ensure it worked as we said it would. We submitted our application to the San Antonio Technology Accelerator Initiative, which is the regional center for the Texas ETF in San Antonio, with our progress to date at that time. We were accepted to present to the State Committee and received an investment award of $1 million. The first tranche was for $250,000, with milestones to have modifications made by the original equipment manufacturer, per our specifications, to an existing off-the-shelf handheld fluorimeter, and to confirm that our assay works by an independent lab, and to develop 3 additional prototype assays. We completed those milestones using an independent USDA-approved lab, and they confirmed that our synthetic DNA assay and flash reader system provided accurate test results within 15 minutes and without the need for culture enrichment. The detection limit they confirmed was 10 bacteria per milliliter with the Campylobacter assay and flash reader. We then received the second tranche for $750,000 to complete the handheld fluorimeter modifications with the OEM with our name and logo, and development of the flash reader system and assays toward commercialization. We now have 5 prototype assays for E. coli, Salmonella, Campylobacter, Listeria, and sugar toxins 1 and 2. We are completing the independent testing and lab validation of the 5 other assays with our internal funding from the Texas ETF to move forward with completing product development of the OnsiteFoodCheck System and flash reader. During the completion of testing, we are also developing our sampling and testing protocols and processes in our standards documentation, with consulting support from the independent USDA-approved lab. This will be important to have completed before we begin our testing with the AOAC for their certification, and to present to the USDA and FDA to make them aware of our testing system. A need-to-know point for potential investors is that food safety testing systems do not require going through the FDA approval process, but we do want to present to them so they know we are moving toward commercialization. That reminds me of a little story that happened about a year ago. I was talking with one of our business partners, Dr. Wayne Shore, discussing an upcoming presentation. I was reading the following: the easiest thing would be to have a portable device that inspectors could carry with them. They could take a tomato, pulverize it, inject the juice into the device, and get an answer in a matter of hours. That would be the Holy Grail. Wayne said, that sounds great, but can you really say it's like the Holy Grail? I said, no, I didn't. I was reading from an Associated Press article on March 10, 2009. That was a quote from Dr. David Atchison, FDA Assistant Commissioner. But I sure do want to pulverize a tomato and inject the juice into our flash reader device in a presentation, because we can get an answer within 15 minutes, and we want to demonstrate that. That brings up another point. Our current name, Pronucleotein Biotechnologies, does not convey what we do, and we need to change that. We ask our lab technicians and others in our affiliate companies, and the team wants to change the name to OnsiteFoodCheck Systems, because that's what we do. We provide a testing system for food and food products that is portable and handheld, and samples can be tested on-site at the sampling location anywhere and at any time. Now that we have confirmed with our first prototype assay that our OnsiteFoodCheck system does provide quick results, and we have 5 other assays that are starting testing and lab validation, we are ready to present to potential investors. Our timeline and planning efforts support our goal at this time to introduce the flash reader system and assay testing platform to the market within 7 to 10 months, and our forecasts show a very good potential within the first year of commercialization. Our first round is for $2.5 million to take to market and commercialize the flash reader system. This funding will be used to build the sales and marketing group, to start to develop relationships and formalize strategic partnerships and sales channels, to staff the business office, to stock the beginning inventory, and to have sufficient working capital until we reach positive cash flow. With the sales volume and profit margins we forecast, we project positive cash flow within the first 6 months.